I just want to summarize it's like the mechanisms, the four major mechanisms of enzyme catalysis. And enzymes can use a variety of mechanisms in order to catalyze it's like the reaction, or in order to convert one substrate into the corresponding product. Here you see it's like the four major mechanisms of enzyme catalysis, which are called the proximity effect, the acid-base catalysis, the nucleophilic catalysis, and the metal ion catalysis. The proximity effect is also called the catalysis by approximation, and it means that it's like that it includes the reaction of two substrates, and you have to bring these two substrates closely to each other in order to reach this enhancement in the rate, and in order to perform this reaction. So it's like there's the substrates, they have to take in a certain a proper orientation in order to be coupled, and also in order to be transformed into the product. One of the biggest mechanisms in enzyme catalysis is acid-base catalysis, and therefore amino acids in the active site of these enzymes take over the role of water, and therefore either donate protons, or they also accept protons. Furthermore, we have some amino acids which can be acting as nucleophiles, and therefore can undergo covalent catalysis. For instance, serine. Serine is a good example of cysteine. It's a good example of a nucleophilic amino acid in the active site which can act as a nucleophile, and therefore can perform a certain chemical reaction. And then some of the cofactors we were mentioning before are metal ions. Most of the enzymes contain metal ions as cofactors, and therefore we have a huge impact of metal ion catalysis in enzyme catalysis. And these metal ions can serve as electrophilic catalysts. They can stabilize negative charges on reaction intermediates. So they are very useful for the enzyme catalyzed reaction.